Thank you very much for the invitation and the opportunity to talk about the research we do. Today, I'm going to talk to you about chemical sensors for biological applications. I am associate professor at the chemistry department. I got my Ph.D. from University of Campinas in Sao Paulo, Brazil, and I have 2 postdoctoral degrees, one from Purdue University and the second one from University of California in San Diego. Some of the achievements I got here at UCF are the RIA and the TIP, and the excellence in undergraduate teaching at the college and the university level. We also had an international exchange program for students from low-income families, so that's also what we do. In the nanobioelectrochemistry lab, we develop chemical sensors with optical and electrochemical transductions. Yes, we make use of nanomaterials, and most of our applications are toward bioanalysis. Our mission is to develop new materials and new methodologies for future generation of sensor platforms. Today, I'm going to talk to you about a few research that we do, being the first one, the visible light activatable, controllable, and robust ion sensing platform using metastable state photoacids. What are photoacids? Photoacids are a compound that liberate a proton upon light irradiation. In fact, we have the photoacid generator, which liberates the proton, but it doesn't regenerate. We also have super photoacids, which they regenerate. However, the proton that they liberate lasts just few nanoseconds. The uniqueness of the metastable photoacid is the fact that the proton that is liberated lasts in order of seconds, minutes. The metastable state photoacid structure was derived from the spiropyran. And the difference with that is that the spiropyran the spiropyran changed the structure upon UV light, but the metastable state photoacid does under visible light. And the most important thing is that the proton that liberates lasts longer, so it can be used for different processes, such as ion exchange process, for example. This compound was proposed in 2011 for the first time, and what we did is to use this compound toward sensing applications with optical transduction. Here you can see the absorbance spectra for the both structures, open and closed. And then here you can see that the sensor can be activated once this compound is included in the sensor. It can be activated around 500 nanometers. Here we show different cycles. And as I mentioned, the most important thing towards this optical sensing is that by including this compound, now we can make the sensing polymeric membrane activatable, so the analysis can be done in the target area. In this area of research, we show that we can use this compound in activatable sensors for detection of calcium, for example. In fact, we developed a library of these compounds, and then we show different applications. Here, we also show that by changing the chemical structure, the functional groups of this compound, we can actually tune the response of the sensor. In order to improve the limit of detection, the sensitivity of our sensors, we also isolate this with fluorophore, in this case, PODP, where we also got good results. Currently, one of my students was able to modify the structure and attach to a gold substrate. So we can study the interface, the charge at the interface, and we actually got a potential in the open circuit of more than 200 millivolts change. In collaboration with our colleague at the engineering department, we also are studying the tunability of these sensors in different kinds of structures, like, for example, microstructure, using these photoactive compounds. A second research area that I would like to talk to you today is about the flexible ion sensors that we do towards wearable applications. One of the biggest challenges for wearable application is the reproducibility of the sensors, and we work towards that. Our flexible platforms are mainly based on paper-based sensors, but we also use plastics substrates. So we develop ink, what we call aqueous ink, that we may use, and then we make conductive our filter paper, right? And upon the making conducting the paper, we put a conducting polymer, poly-302-thiophene, and then we make our sensor. Recently, we improved our ink, and then we developed our organic ink, where we combine the poly-302-thiophene with the carbon nanotubes. Thus, we simplify the preparation of our sensors. The most important observation is that here, as you can see, the contact angle experiments in the aqueous ink that we use, the drop of water lasts for a couple of minutes, but here when we use organic base, so we obtain very highly lipophilic substrate. This helps us to improve the transduction between the substrate, the flexible substrate, with the polymeric membrane. And this is very important so we can obtain very highly reproducible sensors. So you improve from the aqueous ink to the organic ink. So now our sensors doesn't need to be calibrated. In this area, we did various contributions as we showed that we can detect nanomolar limits of detection for different ions. And we also show multiplex analysis. And since we aim to manufacture this kind of sensors, we also need to improve the preparation of these sensors. Thus, we try to avoid preparation steps. Here is our recently paper where we publish the highly reproducibility that our sensors can reach. Currently, we are applying these sensors in the analysis of sodium and potassium in artificial sweat. Here you can see the response from low concentration to high concentration and vice versa. Here is how small our sensors are, and we always work in triplicate, and we also develop a reference electrode, which is in paper-based as well. Here are the selectivity for different ions of our sensor, and this is the response of sodium and potassium for 3 sensors at the time in artificial sweat. Lastly, I would like to talk about our area of research of biosensors for nucleic acids. What is the motivation in this area? We want to develop a universal electrochemical biosensor for detection of DNA, RNA, and microRNAs. Why? Because that will allow us the identification of virus, bacteria, or biomarkers such as a microRNA for cancer disease, for example. Why to detect DNA? Okay, let me tell you that the first time that was possible to quantify DNA was in 1996 by Kramer, where he proposed a molecular beacon probe attached to a fluorophore quencher, which upon target hybridization, the quencher was far enough from the fluorophore so fluorescence can be measured. This approach was adapted to electrochemical transducer in 2003 by Fang, where he, instead of using a fluorophore, he attached an electroactive compound that can be oxidized and reduced, consequently transfer electrons, right, into the probe, which he called DNA stimuli probe. And upon target hybridization, the electroactive species will be far from the electrode surface, so electron transfer is hindered. Although this approach was really good, the disadvantage is that for each target that you have, you will need a new probe, okay? And the other disadvantage was that it was very hard to differentiate one single mismatch. So we propose the multi-component detection probes, which have a universal stimuli probe, and we add 2 intermediate strengths, where one of the intermediate strengths has the electroactive species, which upon target hybridization form the structure of what we call 4-way junction, okay? So you could ask me, why do you complicate your life with those 2 additional strengths? But let me tell you that this approach has many advantages, as, for example, selectivity, because the stabilization of this 4-way junction is shared between 2 strengths. So now we are able to distinguish one single mismatch. Also, there is an improvement in the sensitivity since this previous sensor that I showed you was a signal off because it went from signal, right, to suppression of the signal. And now with the four-way junction, since the electroactive species is in the intermediate strength, so you start from no signal to a signal upon target hybridization. So now we have a signal on, consequently improve the sensitivity of the sensor. And the most important thing is the detection of secondary structures. As you can see, if you have a stimuli probe and a target which has secondary structures, so the thermodynamically is not favor thermodynamically this reaction, right? So, but with the four-way junction, if you have the linear strength, this helps to unwind the probe and the target. So there is a thermodynamically favor this formation of the four-way junction. Here are some of the contributions we did in this area as we show the detection using this platform for microRNA 122. We also use impedance to improve the limit of detection. In this contribution, we were able to detect 0.1 picomolar of the nucleic acid. And here we show the universality of our sensor by using the same probe and being able to detect Zika, and dengue virus by regenerating the sensor. Here is our last publication where we affinate our electrochemical detection with an isothermal amplification technique of the fragment of the Zika virus, and the whole methodology was able to do in less than 1 hour. Currently, we develop a sensor for COVID-2, and the most important thing here is that we can do the analysis in 1 drop of sample, okay? And then we are doing a real sample analysis now. And our recently founded is to optimize the detection of HIV viral loads, where we are going to pursue multiplex analysis as well. Okay, so here with this, I would like to acknowledge all our collaborators here at UCF and also outside of UCF and on other countries, and the agents, the funding agents that support our work. And finally, I would like to thank all my students that were working with me since I started my position here at UCF. And thank you to all of you for your attention. Thank you very much.